In this part of the lecture, I will describe the methodology that can be used to address the type of questions that involve both neuronal level and the level of cognition. And as an example, I will use the facility that we have recently built over the last 7 years or so, which is called the Awake Rodent Research Platform, and it's a core facility, imaging and electrophysiology core facility at the Neuroscience Center at the University of Helsinki. As you can see, it brings together a variety of methods, notably behavioral methods with the brain imaging and electrophysiology recording methods, which allows us to study individual neurons and neuronal ensembles at the same time as we study the mouse behavior, which is tracked automatically during the same experiment. This slide shows some pictures from the core facility. Here on the left, you can see the two-photon microscope, which is a special type of microscope that allows cellular and even subcellular resolution in the brain of a awake and behaving mouse. Then next, you can see here the setup that allows imaging a whole cortex at the same time at lower magnification than the two-photon, but it allows studying the connectivity between different parts of the cortex, again during the same behavioral experiments. In these pictures you can see that the mouse is head fixed to the frame of the microscopes for electrophysiological recordings and also for the imaging experiments. And the same head fixation apparatus is used in most of these setups, which means that we can move the mouse between the setups during the same experiment, and we can compare the neuronal signaling at different scales. For example, we can start with a wide field imaging, with a cortex-wide imaging, identify the areas of interest, and then zoom in with a two-photon microscope, or even perform an electrophysiological recording in the area that was identified with the wide field imaging. There's a variety of behavioral experiments that we can do while recording or imaging in the brain. And one of the readouts is the locomotion, the speed with which the mouse is moving, the trajectory, the place in the mobile home cage where the mouse finds itself. And this all can be analyzed on the fly automatically using the set of magnetic sensors that is built inside the mobile home cage. And the trajectory of the mouse is analyzed on the fly, 100 times per second with a 1 millimeter precision, which gives us the exact information about the mouse's behavior at any given time. To the setup. And instead of just an open maze floating cage, we can also have the mouse walk in the special floating mazes. For example, the T-maze, which is shown here, allows you to test the mouse as it makes a decision whether to turn left or right. The radial maze allows the mouse to run in circles and pay attention to the landmarks that are positioned on the walls. And there are also other types of cage that I will show in the next slides. Another important part of the setups is the liquid reward system, which allows training the mouse automatically to perform certain tasks and be rewarded with a drop of sweet water for performing the task correctly. Also, it can be supplemented with an air puff system that can be positioned on the other side, which will deliver a puff of air on the mouse's whiskers as a means of negative reinforcement when the mouse has performed the task incorrectly. Also importantly, the liquid reward system can be activated directly by the tracking software, meaning that the mouse's behavior translates into a stimulation and reward automatically without a human intervention, which means without a delay and also in the absence of the human investigator in the vicinity of the mouse. which of course increases the reproducibility of these experiments and the robustness of the data. An important part of our behavior, both conscious and unconscious behavior, is the sociability, the ability to interact with other human beings. And for the mouse, it's the ability to interact with another mouse. And as you can see in these slides, the mice can interact with each other while one of them is head fixed, and we can record or image from individual neurons and circuits of neurons in the brain of that mouse, and then correlate it with the kind of interaction that it has with another mouse. We can also supplement the imaging and recording, electrophysiological recording, with ultrasound vocalization recording. And you can see here an ultrasound microphone which is positioned above the animal's head during the sexual behavior, the mating behavior between the head-fixed mouse and the non-head-fixed mouse. And in order to enable this kind of behavior, we have made a special non-transparent roof. The red color is not transparent for the mice, and they feel more cozy in this enclosed environment. This is another type of social interaction where the non-head-fixed mouse is trapped in a jail-like or prison-like compartment, and the head-fixed mouse has the control over the position of the floating cage, and it can come to the vicinity of the entrapped mouse, and they can communicate, and all this communication and trajectory of movement is automatically tracked by the locomotion tracker. And the panels below show that there is a significant difference in these heat maps between the mouse's behavior when there is another mouse entrapped in this enclosure and when there is an empty enclosure or no enclosure at all. More recently, we started using a different design of the cage where the enclosures, there's two of them, and they're attached to a round central hole so that it does not interfere so much with the movement of the mouse. Now, this is another example of the experiment that Claudio Rivera's group is doing. They are performing electrophysiological recordings in the hippocampus, specifically in the dentite gyrus, and the mouse in the meantime is performing the novel object recognition task and also the object relocation task, where the object is moved from one place to another, and the time spent near the new position of the object is measured with the locomotion tracking. I want to show you some examples of the readouts in the brain. One of them will be an anatomical readout, and another one will be functional readout. For anatomical readouts, we are mostly looking at the turnover of dendritic spines, which is the postsynaptic compartment of the synapse, and the large headed spines correspond to functional glutamatergic synapses in the mouse cortex. And here are some examples of how we can image the dendritic spines, the very small structures, around 1 or 2 micrometers in diameter, in the neurons that express a fluorescent protein on a specific promoter, which allows us to visualize not only the dendrites but also the cell bodies and the whole dendritic tree structure. Importantly, for this kind of experiments, the same dendritic spines can be revisited at different time points with several days or even weeks interval. And this is important because the synaptic plasticity is believed to consist in the turnover of synapses. Some synapses are added, other synapses are eliminated. And this turnover, the rate of the turnover, is proportional to the level of plasticity that the cortical neurons possess at a given time. So one can track the turnover of dendritic spines and then test certain drugs that increase or decrease the plasticity or certain traumatic conditions or disease models and make quantitative estimates of the level of synaptic plasticity. So here at the bottom of the slide, you can see the snapshots of the dendritic segment with several dendritic spines on it, and the arrow indicates the dendritic spine that was stable for the whole duration of the week except for the last time point where it disappeared. This is just the animation that shows the same thing, and you can clearly see that there is a couple of spines that are appearing at the later time points during the imaging. Now, it's very important that the mouse stays awake during these experiments, the dendritic spine imaging experiments, because the anesthetics do have a very strong effect on dendritic spine turnover. They do promote, or at least affect, the synaptic plasticity very strongly, as is shown in the next slide. This is a publication from a couple of years ago, which was done in collaboration with a pharmaceutical company, Sunovion, which at the time was developing an antidepressant drug that would have a more rapid action compared to the classic SSRIs, the serotonin reverse uptake inhibitors, and would have a long-lasting effects after a single exposure to an antidepressant. And a good candidate for this is ketamine, which happens to be an anesthetic used in both veterinary and human clinical practice since many years and decades. And it also has an antidepressant effect on especially severely depressed patients with the suicidal thoughts. It reverses the depression quickly within hours or days, as opposed to several weeks of the onset of SSRIs. And therefore, this pharmaceutical company was interested to study the mechanism of action of ketamine on synapses in awake mice. So in this study, which was done in collaboration with Eero Kastren's group here in Helsinki and Sergei Kirov's group in Augusta, Georgia, what we did, we imaged dendritic spines at several time points, and then we treated a group of mice with saline without ketamine, and another group of mice with a ketamine solution. It was a daily injection, and the dose that we used here was sub-anesthetic, meaning that the mouse remained awake during the experiment, but it did see the therapeutic dose, antidepressant dose of ketamine, as indicated by the behavioral experiments, also shown here on the right. The main effect that we observed is that the group of mice that received daily ketamine injections did show twice as many spines formed during a certain period of time as compared to the control group, whereas the spine elimination dynamics was not affected. This produced an increase in the number of functional spines in response to the chronic ketamine treatment, which is a very likely mechanism underlying the antidepressant effect of ketamine, because it increases the plasticity reservoir, the capacity of the brain to adapt to new conditions and stop the patterns that were associated with a depressive state. We also collaborated with Eero's group on another anesthetic, isoflurane, which interestingly did not produce a similar effect on dendritic spine turnover compared to ketamine, which indicates that different antidepressants and different anesthetics can have different mechanisms of action, or perhaps different locations or target areas in the brain of mice.